Hi, good morning, good afternoon, everyone. Thank you for joining me today in this final webinar in the ACEL webinar series. My name is Josephine Lambong. I am the Senior Manager of Science and Industry Affairs at ARM. Today's webinar is on the topic of regulatory considerations for cell-based therapies and will be delivered by two of the key contributors of Chapter 2 of ACEL, who I will introduce in just a few minutes. For those who are not familiar, ARM is the leading international advocacy organization championing the benefit of engineered cell therapies and genetic medicines. ARM builds the future of medicine by convening the sector, facilitating influential exchanges on policies and practices, and advancing the narrative with data and analysis. We actively engage the key stakeholders to enable the development of advanced therapies and to modernize healthcare systems so that patients benefit from the durable and potentially curative treatments. As the global voice of the sector, we represent more than 400 members globally, which includes emerging and established biotechnology companies, academic and medical research institutions, as well as patient organizations. I'm sure everyone here knows that manufacturing for cell and gene therapies is a complex process, and it is often a rate-limiting step in the commercialization of regenerative medicines. We as an industry is in the process of rapidly scaling up, and there are more phase 3 studies and more commercial product approvals anticipated. However, products are being held up at the BLA stage due to CMC issues. So we see that the advances are being made in the manufacturing technologies and the experience in the workforce are overall improving, which expands the process understanding. However, standards and best practices in the field are not advancing as quickly. So this is the pre-competitive area where ARM focuses our efforts in to further advance the process understanding in the industry to facilitate product development and commercialization. ACEL is a project that ARM undertook several years ago and was completed in 2022 after we received feedback from our members about the CMC issues that the field as a whole is experiencing. ACEL is the best practices document which incorporates collaborative case studies presenting the implementation of QBD principles and cell-based therapies. To make ACEL an effective resource and reflective of the ongoing innovation in the sector, we chose to focus on immune cell therapy as the case study, and in particular, further focus the document on autologous CAR-T, given the recent approvals and the number of products that are available to the patients in the market. ACEL follows the success of AGene, the sister project, which focuses on AAV vectors, and AMAB, which was instrumental in allowing advances to occur in the monoclonal antibody industry in terms of improving manufacturing quality, optimization, and not to mention to drive down costs. We are very fortunate at ARM to have a large number of subject matter experts from our member companies, including our two speakers today. There are many expert contributors for this project from more than 30 ARM member companies. And this project is also supported by Nimble, who generously provided funding for ACEL, which allowed us to employ technical writers to support the project. The document is intended to benefit the entire field, so it is available as an open access document in our website at the link provided here. So this is what the contents of ACEL look like. As you can see, it covers quite a big scope. Chapter 2 covers regulatory considerations, comparability, and standard. 3 and 4 are really the foundational principles of QBD. 5 and 6 touches upon the starting materials and ancillary materials considerations. 8 covers the manufacturing process, and 9 analytics. Today's talk will cover the elements of regulatory considerations laid out in Chapter 2. So the webinar today is the final one out of the 9-part webinar series for the ACEL educational rollout. Thank you very much to those who have joined us for this series. Our hope is that ACEL, both the document and the webinar, can serve as an educational tool to the community. And we hope that this document will be used by product developers, educators at universities, and we also have the aspirational goal that this will be used to help educate regulators. And before we move on, just one housekeeping item. You can use the Q&A function in Zoom to ask the questions, and we will try to get to as many as we can at the end of the talk. If we didn't get to your questions, there will be an opportunity for us to compile them and post a response in the webinar website. So without further ado, I would like to introduce our expert speakers for today's webinar. Our first speaker is Michael Mendicino. Mike is a multidisciplinary cell and gene therapy expert with over 24 years of experience, educated in molecular, cellular biology, genetics, immunology, and regulatory science. Michael founded Hybrid Concept International as a consultancy and advisory firm and is the president, owner, and chief consultant and advisor. Hybrid Concept is a niche cell and gene firm that successfully served over 145 clients in the last 6 years. Mike manages over 20 deep subject matter experts in different disciplines under the HCI umbrella, providing clients multidisciplinary leadership or support across regulatory, CMC, quality, preclinical, clinical, and publishing. Let's see, Mike is representative on multiple ARM committees as well as ISCT and is a co-founder and on the board of directors for Standards Coordinating Body. Our second speaker is Emmanuelle Sabbah-Petrover. Emmanuelle is the Director of Regulatory Science for Cell and Gene Therapy Products at VCLS. She manages projects from early stage of development to late clinical phases and preparation of a MAA. Apologies. She provides strategic and regulatory input on the development of ATMPs, working closely with developers on the long term. Emmanuelle has specific expertise and experience in agencies meetings in the EU with more 40 plus scientific advices from EMA or national agencies. And so with that, I will hand it over to our first speaker, Mike Mendicino, to start your presentation.
Yes, thank you very much. Give me a second to share my screen. So thanks everyone for joining. This is ACell Chapter 2 webinar. As mentioned by Josephine, I'll be giving the US FDA regulatory considerations presentation, and Emmanuelle after me on the EU regulatory considerations. Waiting for my slides to pull up 1 second. And just to cover what's in scope for today's webinar, what's in scope again are the US and EU regulatory considerations, and we'll very briefly touch on the main differences between the US and EU. What is out of scope, although covered in the ACell Chapter 2, are the standard section, comparability section, and then, of course, other regulatory agencies and jurisdictions outside of the US and EU. So FDA legislation, regulations, and guidance essentially start with a bill being proposed that is then reconciled in Congress, signed into law by the president, translated or inserted into the United States Code, and then in some cases, the Code of Federal Regulations at a multi-agency level, and then at an agency-specific level, such as at the FDA. This can turn into guidance, standard operating policy and procedures, or other such relevant documents. The most recent and some was covered today is the PDUFA 7 legislation that had numerous commitments that benefit both cell and gene specifically, as well as more generally biologics and drugs. Just to cover a little bit about FDA terminology, the cell therapy is really a pure cell therapy, so like an MSC or an NK cell, not a CAR T cell. A gene therapy can be an ex vivo gene therapy that is cell-based, that is either gene modified or gene edited. There can also be in vivo gene therapies like AAV or like a gene edited product that's administered directly in vivo. A combination product can be a cell or gene that's combined with a scaffold, or it could be utilizing a novel delivery device, which is a little different than the definition in Europe. And medical device, although we won't cover here, are also reviewed in some cases in the Office of Therapeutic Products. Some of the key recent FDA guidance in cell and gene, a couple of important ones that have been finalized, the gene therapy CMC guidance for INDs that go into common technical document specific advice. There's one for studying multiple versions of a cell or gene therapy product in an early phase clinical trial that's been finalized. and that uses the primary and secondary IND scheme in an umbrella trial setting, looking at different iterations within the same disease or condition. Relatively recently, while still in draft form, there's the genome editing guidance and the CAR-T guidance. And ARM, along with many others, I'm sure, have submitted comments to the FDA, and the FDA, I'm sure, is looking at all those comments as they consider finalizing those guidances at the appropriate time. Very recently, the manufacturing changes and comparability for human cell and gene therapy products has come out. In fact, ARM is currently providing comments during the extended comment period, and look forward to see progress on that. There is a CBER guidance agenda. The link is here for 2023. The two I'll call out, other than the comparability I already mentioned, is a potency assurance for cellular and gene therapy products. We very much look forward to that, given the last cell and gene potency specific guidance is from 2013. Also, the safety testing for human allogeneic cells will also hopefully help align the field in cases where an allogeneic starting material is utilized, but there is no master cell bank stage. And the question of the timing and the need for adventitious agent testing and where in the process is definitely going to be helpful for sponsors. For IND and BLA content in common technical document format, you can see on the left-hand side the ICH triangle, 5 modules. Module 1 is regional. Module 2 are the summaries for the 3 main disciplines, quality or CMC, farm tox or non-clinical or preclinical, and clinical. Module 3 is quality or CMC. Module 4, non-clinical. Module 5, clinical. And you can look at the ICH website for more detail. Some general comments I'll make. Investigator-initiated INDs do not have to follow the common technical document format at this current time, although all commercial INDs do. INDs with prior clinical experience would have more CTD sections populated than an IND without any such clinical data, for example, in the clinical summary in Module 2 or potentially in Module 5. And of course, the BLA is much more comprehensively addressing all of the sections, including numerous Module 1 sections that wouldn't be required in an initial IND. Here's just a screenshot of the FDA's webpage for investigator-initiated INDs. The website link is here, not to go into detail other than to say that the infrastructure is more simplified than the common technical document, essentially is broken down other than introductory statement and the required forms by CMC, farm tox, IB, clinical protocols, and other relevant information. Although not so critical to cases of biologic drugs, I did want to at least bring up the difference between a 351 HCTP or human cell tissue or cell and tissue derived product versus a 361 HCTP. This is actually covered quite well in a guidance that was finalized with the link here, regulatory considerations for HCTP's minimal manipulation and homologous use. Essentially, for a product to not have to go through the IND to BLA process, it would have to be no more than minimally manipulated, intended for homologous use, not combined with something else other than very limited exceptions, and not have a systemic effect or dependent on metabolic activity of living cells, or to be autologous or related to or utilizing allo that's related. There are a couple of exceptions to this. Again, it's covered well in the guidance. The reason why I bring this up is in the case of a CAR-T, if you're utilizing an allogeneic CAR-T source, whether that be somatic or pluripotent, there are requirements coming from 21 CFR 1271 that originated in the PHS Act 361 as it pertains to donor eligibility. What is donor eligibility? It contains both donor screening component and a donor testing component. The donor screening component is actually covered quite well at the AABB website with the link here, including a donor screening questionnaire template. And then there's also the donor testing requirement that requires certain human viruses and adventitious agents to be tested with FDA licensed, cleared, or approved kits. And the listing is actually on this website here. Again, you'll get this presentation after to be able to go to all these websites. And it's important that also those tests are done in a CLIA certified lab. Another bucket of considerations are with regards to master files and their use in the USA. While the master file system in the USA is quite comprehensive and allows for many options to be filed to benefit sponsors and their partners in the development process, there are still a couple of outstanding issues that need to be resolved, such as timing of the review of an original master file and the potential risk to the first IND sponsor to cross-reference it. How does one meet with the FDA to discuss a master file when that party does not have an IND in place in a consistent fashion? And then how does the contents of a master file impact the common technical document as it is supposed to be filed in common technical document format? There are many different types of content for a master file. Some examples as it pertains to CAR-T can include the plasmid that is used to manufacture the viral vector or mRNA of the CAR transgene. It could be manufacturing enabling equipment. For example, the electroporator for non-viral gene modification or gene editing of a CAR-T. It could be for the lentiviral vector for the ex vivo gene modification itself for the CAR-T transduction, or even a drug product, for example, in a case where a sponsor is working with a large biopharma partner or for their own internal regulatory efficiencies, cross-referencing a single CMC or master file for its CMC component across multiple INDs. What is regulatory science? So regulatory science is defined as the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of all FDA-regulated products. The reason why I bring up that definition, as mentioned earlier, PDUFA 7 has a bunch of commitments, some of which pertain specifically to cell and gene, some of which pertain more generally to biologics and drugs. A lot of them fall under the attempt at progress within the guise of regulatory science. On the left-hand side, you'll see some cell and gene specific commitments. On the right-hand side, some not cell and gene specific, but relevant for cell and gene PDUFA 7 commitments. Starting at the top left, there is now an Office of Therapeutic Products Super Office that essentially more divisions, more branch offices, more reviewers, more training, essentially to keep up with the exponential growth in the cell and gene space. A second component is leveraging knowledge. For example, in a tool or a platform used across multiple cell and gene products, which in fact got taken another step further in the Omnibus spending bill in 2022, establishing a platform designation that's forthcoming. While of course there's a patient-focused development for cell and gene, specifically in the commitment letter, there's a lot of other patient-focused development aspects for general biologics and drugs. One thing I'm personally looking forward to is an update on the RMAT designation program progress for cell and gene that's mentioned in the PDUFA commitment. On the right-hand side, for those of you that submitted Interact when it was considered informal, now it's been formalized in the FDA SOPP. The links are all down here. There's now a CMC development and readiness pilot program that's currently active, which addresses one of the issues that sponsors have been facing for quite some time, which is the how to accelerate CMC development in the advent of accelerated clinical development in the granting of a designation such as an RMAT or a breakthrough designation, breakthrough therapy designation. There's a STAR pilot program that essentially is meant to provide efficiencies in the supplemental BLA process for a product that's already had a marketing authorization in one disease or indication and is filing to have a approval for another indication. And then a Type D meeting, which is also in the FDA SOPP formal meeting document, and it essentially provides yet another meeting type that is more focused in the number of disciplines and questions it allows, and for example, could focus on comparability or potency or some other important element of development. Here's a regulatory pathway for cell and gene as documented in ACell. Of course, it's hard to develop one schema that flows for everything. The three-phase paradigm, of course, has been moved away from for quite some time in a lot of the cell and gene currently being developed. Some of the points I just wanted to make really are stated here. The granting of expedited program designations and our use of innovative trial designs, such as adaptive designs, may lead to changes to these development milestones or other interactions with the FDA, such as the need to accelerate CGMP compliant commercial manufacturing and CMC activities in the case where there are only 1 or 2 trials utilized to support licensure. In these cases, early and significant product and process characterization and early and consistent FDA interaction are among critical tasks to ensure agreement on CGMP compliance and CMC activities for licensure support. And I'll just reference back to my prior slide, and the numerous PDUFA 7 commitments actually help support exactly that. There has been significant updates to the FDA regulatory meeting and interaction since ACell, including the formalization of Interact, the Type D meeting. So instead of using that figure, I took this directly from the updated FDA SOPP. The link is here. Just to point out again, the new parts: Type D meeting, 14 days to get an FDA response, submit your package with your meeting request, and the meeting, which could be a WRO or written request only, is within 50 days. Interact, now formalized, still packaged with request, 25 days to FDA response. The meeting, such as the telecon, within 75 days, and the preliminary responses received may be annotated based on the output of, for example, a telecon. There's also a Type C meeting specifically for early consultation on the use of new surrogate endpoints, which of course could be very important for an RMAT designated sponsor. The ACell chapter goes into a bit of detail with regards to what to put into what drug substance CPD section or drug product CPD section. We won't have time to go into detail in this webinar as it's well described in the chapter, but just to provide a couple of examples for context. An example of a pure cell therapy CMC CPD can be quite simple in comparison to the next example. You have your cell-based drug substance section set, S1 through S7. Again, that's described in the CTD guidances that both FDA and ICH follow, and is also covered in the gene therapy CMC IND guidance finalized in January 2020. The control of materials, or section 32S23, would contain the donor eligibility info if allogeneic. And then of course, there's a drug product section set, P1 through P8. Here's a more complicated example. So you have a gene modified through lenti and a gene edited through RMP cell-based gene therapy product, such as an allogeneic gene edited CAR T. In this case, you'll have additional section sets as part of the CTD. At least this is the way that I would be approaching it with my clients. So in addition to the cell-based drug substance section and the drug product section, there would be a separate drug substance section set for RMP, which in its control of materials section would contain information on a guide RNA and Cas9, for example. Another drug substance section set for the lentiviral vector, and in its control of materials cover the plasmid bacterial master cell bank and then the mammalian master cell bank for the lentiviral vector. The FDA expedited regulatory program designations review or approval pathways, again, is well covered in the ACell Chapter 2. I'm not going to get into details here, just to say that there's been a progress between 97 and 2017 with the number of designations that have become available, and RMAT is specifically available to that which is regulated at the Office of Therapeutic Products in many of the cases. In fact, the definition of what is considered a regenerative medicine product has expanded over time since it was originally established. So what are some of the differences between the fast track designation, breakthrough designation, and RMAT? Generally speaking, fast track you can get with potentially even just non-clinical data. Breakthrough therapy requires preliminary clinical evidence that indicates a cell or gene product may demonstrate substantial improvement, whereas the regenerative medicine and advanced therapy or RMAT designation just requires preliminary evidence that indicates the potential to address unmet medical need. So the clinical bar is lower for RMAT, but there's more advantages. Additional advantages for RMAT over breakthrough include potential ways to support accelerated approval and satisfy post-approval requirements. Generally speaking, you'd want to file for these either with your IND or no later than your EOP2 meeting to actually really get any benefit outside of a press release. And another thing to note is the designation can be rescinded if for example, the clinical data that was filed that allowed for the grant in the first place was done with an earlier iteration of CMC than the later stage clinical development, and comparability was not met, which is a very important consideration. You can actually get the FDA RMAT requests granted, denied, and withdrawn numbers right from the FDA website at this link. And you can see the numbers have generally increased over time, and the number of granted versus denied has been relatively stable. For FDA rare disease programs, I found a publication recently by some of the folks at NIH that in human gene therapy that actually covered this very well. So I figured I'd just put the link here for you all to reference later, and all of these links work within the presentation to cover the two main programs, which is the orphan drug designation and the rare pediatric disease designation. There's a lot of detail in there that I won't get into, but you can file both of those prior to even having an IND. And although they don't have the benefits of the other expedited programs that was mentioned earlier, there are other benefits that they have that the expedited programs do not have. And you can find all this on the FDA website. CBER actually has an advanced technologies team at the CBER level that is not only for cell and gene therapy products and associated tools and technologies. This allows potential product or tool developer to meet with the FDA to discuss non-product specific and indication specific issues. It's been around for a while. The time investment is not that great. It's a 2-page application. The progress within CBER CAT is something that I'm definitely looking forward to, as it was a commitment within PDUFA 7 to explore the historical value of this program and where it can go, and potentially how it can contribute to the platform designation process that's forthcoming. For an example of optimizing CAR-T US development, let's just say in a perfect world where you're eligible and granted all the designations and things happen in the right order and the data looks great, an optimized pathway could be you're looking at a rare disease indication, so you get your orphan drug designation, you file your IND, it's allowed to proceed, you get into your first in human trial, you have the appropriate preliminary clinical evidence, and you're otherwise eligible for RMAT. You submit an RMAT, get the RMAT designation. You have the subsequent meetings with the FDA. That there is a potential for including the discussion of a surrogate marker approach for accelerated approval. That could potentially go through a special protocol assessment with the FDA to ensure the the pivotal trial or extended trial design is concurred by the FDA. Of course, the data would be a review issue, then submitting for accelerated approval. And if you've incorporated, if your product has incorporated an antigen that could potentially target multiple cancer indications, and the trials designed were appropriate and the data was appropriate, you could potentially file a tumor agnostic label for a tumor agnostic label, and with the expedited program granting, try to utilize rolling review, which is submitting the modules as they are completed instead of all at once, and then priority review, which is shaving basically 4 months off the BLA review timeframe. A couple of key issues, again, these are topics of their own, just to bring up. One, of course, is comparability. As mentioned, there's a draft guidance that just came out. In fact, one of the FDA colleagues has actually done a webinar for the draft guidance, which is actually quite helpful and not all that common for draft guidances. Things that are covered in there include the risk management, planning for future changes, phase-dependent expectations, comparability study content, and then how one can obtain advice. The webinar is here. And as I mentioned before, ARM is going to be doing a commentary on this draft guidance, preparing it now. And then, of course, potency, another favorite topic. ARM, along with ASGCT and the FDA, had a scientific exchange, which culminated into a white paper that's a link here, covering many of the issues sponsors are experiencing today in the potency world. Some of the things I think that, going back to the example of the allogeneic gene-edited CAR T, perhaps also with a cytokine included in the transgene in the viral vector or non-viral vector construct. For drug substance potency, when in development, and how does one approach these different elements? For example, the lentiviral vector, the RNP, or other. At the drug product stage, one can look at CAR T activity in vitro, but what about the contribution of the cytokine transgene, or the contribution of the hypoalloreactivity due to the gene editing? So again, hopefully between the finalized guidances that are coming for genome editing and CAR T and the new guidance, draft guidance and potency assurance, we'll be getting towards addressing some of these issues. With that, I turn the panel discussion over to Emmanuelle to discuss the EU regulatory considerations. Thank you for your time. I'll stop sharing, and Emmanuelle, please go ahead.
Yes, thank you, Michael, for the nice presentation. I will now focus more on the development of advanced therapy medicinal products in Europe, focusing on regulatory aspects and not that much on the CMC ones. So first, a little bit of background on the legislation of ATMPs in Europe. These products are governed by two main texts. One directive on the human medicinal products that was released in 2001, and the second document is the ATMP regulation that was released in 2007. Both documents will provide you with the definitions of ATMPs, but also on gene therapy medicinal product, somatic cell therapy medicinal product, as well as tissue engineered product, which is a difference with the US, and combined ATMP. The directive on the human medicinal products also introduced the notion of the risk-based approach for ATMPs, and the fact that the marketing authorization application, MAA, must go through the centralized procedure, i.e. via the European Medicine Agency, and not at national member state level. The ATMP regulation introduced the Committee for Advanced Therapies, the CAT, at the EMA, which will be replaced as a working group and not as a committee anymore, which is under discussion, and introduced specific procedures that we will present in a few minutes, offered by the EMA. As mentioned by Michael, on the US side, in Europe, we also have a lot of guidelines to support the development of ATMPs that you can find on the EMA website and that are classified per type of product. We also have GMP and GCP, good clinical practice and good manufacturing practice for ATMP, both documents that were released a few years ago already. Now, if we go into a little bit more granularity to the definition per se of ATMPs, one thing that is really important and to bear in mind is that these products, as I said, are human medicinal products, but also should be of biological origin, meaning that they should be produced or extracted from a biological source. And this is key to establish the formal classification. For instance, one recent example is the mRNA COVID vaccines that are not considered as GTMP in Europe. And that's why they have been assessed for marketing authorization by the CHMP, the Committee for Human Medicinal Products, and not by the CAT. We then have the definition of a somatic cell therapy medicinal product and how we should classify a product originated from cells between a somatic cell therapy medicinal product and a tissue engineered product. It's mainly related to the primary mode of action, while a cell therapy in Europe will have primary mode of action in the view of a pharmacological, immunological, or metabolic action. A tissue engineered product is meant to regenerate, repair, or replace a human tissue. Importantly, both for cell therapy and tissue engineered product, the cells have to be substantially manipulated, and the list of these substantial manipulation is included in the European directive. We then have a combined ATMP, which is any of the three that we briefly presented, combined to a medicinal product. Now let's go into discussing a bit the interactions that you can have from competent authorities in Europe. First, scientific advice, which you are all probably aware of. So most of the competent authorities in Europe do offer scientific advice, meaning at national level by national competent authorities or by the European Medicine Agency, the EMA. In order for a developer to decide whether to seek advice from EMA or at the national level, then it's basically on a case-by-case basis and should be aligned with your overall clinical development. One example, if you are already planning to conduct a clinical study in Europe, then you may want to seek advice from the national agencies of the countries where you intend to perform your clinical study as a way to test the water, if I may say so, to see whether they would accept an upcoming clinical trial application or if there are already some hurdles that would be difficult for you to overcome. On the other hand, if you have questions on the overall development of your product, then you may want to seek advice from the EMA because it's the agency that is competent to assess your future marketing application. For scientific advice at national level, the procedure is the following. You have to submit a meeting request form that will include your questions and propose meeting dates. And then once the date is agreed with the agency, you will submit the briefing document, usually a few weeks ahead of the meeting, and then the presentation. The meeting will take place, usually 90 minutes, and you will have to submit meeting minutes. Scientific advice at the European agency is slightly different. you have to submit a draft briefing document along your initial meeting request. Then there's a validation period, usually of 3 weeks, and you may be requested to slightly amend your briefing document. Then the procedure will start, and it's up to the EMA to invite you to a discussion meeting or to provide only written responses. The duration of the procedure is hence of 40 or 70 days, depending on whether you are granted a meeting or not. And in both cases, you will get a final advice letter from the EMA with the advice. There are fees to pay for scientific advice, except in some European member states. And we strongly recommend you submit several scientific advice requests throughout your development. What we have noticed, based on our experience, is that the development of an ATMP usually requires more scientific advice throughout the development than a small molecule. And the agencies will always be happy to support your development and at the same time to gain knowledge on the scientific advances. On this slide, you can see a summary of the main differences between scientific advice from EMA or from national agency. I won't further comment for the sake of time. What I would like to introduce to some of you who may not be already aware of is to the simultaneous National Scientific Advice SNSA procedure. This pilot program was launched in 2020, so quite recently, as a way for a developer to submit a meeting request, a scientific advice meeting request, to 2 national agencies simultaneously. A third agency can act as an observer, but won't further comment and won't provide final advice. The procedure is coordinated by the Paul-Ehrlich Institute, a competent authority from Germany, and there are fixed timelines, contrarily to national advice as a standalone procedure. You have here the list of the participating agencies. It's on a voluntary basis. And because there are stringent timelines, you have to submit a request, and then the agencies will accept or not being included in the SNSA procedure. Another type of meeting that you can get from the EMA, besides the scientific advice, is the Innovation Task Force meeting, the ITF meeting. As I said, it's offered by the EMA, and it's usually meant for innovative medicines or new methods or technologies. Contrarily to scientific advice, there is no fixed timetable, and it's free of charge. However, we would say that it's less formal than a scientific advice, and you won't get final advice letter, and the minutes of the meeting are the ones that are issued by the company and reviewed by the ITF. As for a scientific advice, you have to submit a request, and then it will go to a kind of a triage at the ITF, at the EMA, who will accept or not to grant you an ITF meeting. What we have seen is that sometimes the ITF will consider that the questions that you are raising are more for a scientific advice procedure, and they will redirect you to a scientific advice. If granted, you will have to share a briefing document. There's a specific template as for scientific advice. Submit the presentation of the meeting ahead of it, and then minutes that will be reviewed by the ITF. ITF can be requested as a scientific advice anytime during the development of a product. Now, in addition to consulting with competent authorities, we wanted to draw your attention to the fact that engaging with health and technology assessment, HTA bodies, is crucial, especially in Europe. Why? Well, the reason is that the EMA will evaluate the safety and efficacy of your ATMP, while the HTA bodies will evaluate the relative effectiveness and the cost effectiveness of your product. Hence, EMA and HTA bodies may not have the same expectations or view on critical aspects of your development, such as the unmet medical need, the target population, the pivotal study design, including comparators or endpoints, and etc. One thing to bear in mind is that from January 2025, HTA bodies will evaluate ATMPs via a joint clinical assessment on the clinical domains, while other domains will be left for evaluation at national level. Now, as Michael mentioned, the 3 designations that are offered by the FDA in the US, including the RMAT that is specific to regenerative medicine, I wanted to briefly compare it with the PRIME scheme offered by the EMA. The PRIME scheme is quite recent. It was implemented in 2016 by the EMA as a way to mimic the designations already offered by the FDA. If you are granted the PRIME scheme, then you will have an early appointment of an EMA rapporteur from the CAT for ATMPs, which will be the person leading the assessment of your marketing application afterwards. You will also have an EMA dedicated contact point to ease the daily interactions. Other key features that I won't discuss today. To be eligible to the PRIME scheme, you have to demonstrate an unmet medical need as well as the potential to address this unmet medical need. Importantly to note that the ATMP have the highest success rate of PRIME eligibility requests. Procedural aspects are as follows. You submit a PRIME eligibility request using the briefing document template from the EMA, and then there's validation and 40-day procedure from the D0. It's free of charge, and you can apply at various stages of development depending on whether you are an academia, have the small and major enterprise stages from the EMA, or if you are a biotech. Now, after having discussed general aspects of regulatory development in Europe, let's discuss 3 points that are specific to ATMP. First, the classification procedure that is offered by the EMA. It's intended for developers who are unsure of the classification of the product, whether it fulfills the eligibility criteria to be classified as an ATMP, and if so, if it's a gene, cell, tissue engineered product, or a combined ATMP. It's not mandatory, it's really optional, and can be requested until the marketing authorization application. However, we would recommend you submit a formal classification when you are unsure, and especially if you are dealing with cell-based products. Why? Because then you can adapt and adjust your development to the classification. It doesn't have always a huge impact, but if your product is classified as a cell therapy or a tissue engineered product, then your non-clinical or clinical endpoint might be slightly different, depending on whether you are, depending on whether you need to prove metabolic activity or an immunological activity, or replacement of a tissue, for instance. As for any procedure with the EMA, you have to submit a package that will be validated and the procedure will start. The CAT will issue its formal recommendation within 60 days, and it's free of charge. The second procedure that is specific to ATMP is the certification procedure. it's not widely used, so not very common. In order to apply for the certification, you have to submit modules, quality modules or non-clinical modules, and then the EMA, the CAT, will assess whether the data met the requirements that are applicable for the evaluation of a marketing application. So you won't get scientific discussion at all, but kind of a stamp on the data and on the modules that you have submitted. Again, the classification is optional, and it's restricted to companies that are developing ATMPs with the SME status. Last but not least, something that we wanted to mention, the fact that some of the gene therapy medicinal products are genetically modified organisms, GMOs. For instance, all the vectors derived from viruses. As such, they must comply with one of the two European directives, either on the deliberate release or on the content used of GMOs. That means that there are specific documents to prepare and submit ahead of manufacturing or use in non-clinical or clinical studies of these products in an effort to harmonize the procedures, because they are still at national level, even for clinical trial applications. There are some forms and good practice documents that are being published for some of these products. As concluding remark, we wanted to highlight that there are less than 20 ATMPs that have been approved so far in Europe, and some of them have already been withdrawn from the market, mainly for commercial reasons. It's really important to keep up to date with the regulations and the guidances. It's crucial to get input from regulators throughout the development, via scientific advice, for instance. And it's equally important to meet expectations from the payers, so to meet HTA bodies, and also more and more important to involve patients through association or representative. And with that, I will hand over to Michael, who will present some key differences.
Thank you, Emmanuelle. So at a very high level, there are numerous other differences not mentioned, even terminological differences as discussed earlier, but some of the buckets that are different between the US FDA and EU EMA, the master file system. So there's a comprehensive system in the US, there isn't such a system in Europe. The experience can be National Competent Authority or NCA case specific, and there may be some no-goes versus where in the US that something like that could be covered in a master file. With the advent of the clinical trial regulations in Europe, it's unknown the impact that might have on the progress towards the master file system in Europe. As I mentioned in my part of the presentation, the donor eligibility requirements as it pertains to the US under PHS Act 361 or 21 CFR 1271 have a donor screening and donor testing requirements that are not 100% aligned with other jurisdictions, including Europe, which can actually prevent, for example, a European donor from being used in the US without going through an exception or alternative application that the FDA does have an option for. Another difference is the early clinical development CGMP compliance flexibility that the US FDA has. There is a phase 1 GMP guidance. And then as Emmanuelle just covered, there's a GMO application requirements in the EU, whereas there is not in the US way back when there was an NIH RAC or RAC that no longer exists in the same, that has that requirement any longer. So it'd be interesting to, to understand the additional burden that a sponsor may have going to Europe with the GMO application requirements. Please, next slide. So we hope you all now have a better understanding of the regulatory environment for cell and gene-based products in the US and EU separately, and a little bit about the differences. There are a lot of efforts towards convergence, and that's, again, another topic for a whole webinar. But due to these differences, a sponsor should consider the chronological order and the timing of which one might go from one region to the other, or consider a global strategy, either for clinical development or approval. Thanks for listening, and we're happy to take any questions.
Great. Thank you so much, Mike and Emmanuelle, for the fantastic talk. So I'm going to go ahead to the Q&A that I think we've received a few questions from the audience. The first one, I'm going to direct it to Mike. Is master file in the US accepted on the IND or also at the BLA stage?
That's a very good question. So master files can be cross-referenced with a letter of authorization in an IND. My general understanding is it at the bare minimum significantly frowned upon, if not allowed at the BLA stage. But that's not there's no single answer to that from my understanding. Part of the issue is, and the reason why, in my understanding, Europe doesn't have such a master file system, is because the conceptual differences in thinking with regards to what the sponsor or what the product developer should have access to with regards to the tools and technologies that it's using in its manufacturing process, and the ability to do process development, process evaluation, process validation, analytical development, and validation all can be challenging when there's significant components that a sponsor does not have access to, which is why I think later in development, it would be prudent for one to consider partnering with a tool or technology provider that has a master file in a way that would allow more free flow sharing of information with that specific party, versus relying on the master file cross-referencing exclusively, just in case there are challenges as you get to late stage development and the BLA stage. But I'm not speaking for the FDA here, obviously, and there may be a better answer coming from, or would be most likely a better answer coming from one of them. And I definitely suggest that you take that up in an FDA meeting and ask that question as it pertains to your specific product and your specific case.
Great. Thank you, Mike. Yeah. The next one for Emmanuelle. Does the EMA accept a US only study for an MAA in the EU?
Difficult to answer. The EMA could accept, but you'll have to discuss ahead of the submission of the marketing authorization with the agency, and also demonstrate that there are no crucial differences in terms of the target population, the standard of care, etc. And as such, that US population can represent EU population as well. And this is usually the kind of question that we include in a scientific advice for a product that is at a late stage of development.
Great. Thank you, Emmanuelle. For both of you, what is the difference for hospital bedside use of cell and gene therapies between the EU and US regulations?
Well, I mean, I don't know if you want to speak to the hospital exemption, Emmanuelle, on this topic. It would probably better come from you and the difference there.
Yeah, so hospital exemption is specific to Europe indeed. But here, I think the question is about the use of CGT that are maybe manufactured at the patient bedside. So I won't be able to comment on those aspects in terms of what is expected. Again, in Europe, every step of the manufacturing process should be under GMP. So if you implement a step that is not considered as substantially, I would say, impact your product, then it may be accepted, but such as throwing a bag with cells or things like that. But if you put the cells in culture, it will be considered as a GMP step.
Yeah, I would just add, if the question was referring to the decentralized manufacturing approach, that's becoming more and more common. And some of the considerations, of course, are common use of SOPs, whether it's at the biologic starting material stage or at the with regards to the manufacturing process itself, in order to assess the ability to leverage pooling data as you further along in development. But it's complicated for sure.
Thank you both. Time maybe for one more question. Is there a regulatory expectation on the number of PPQ batches required for cell and gene therapy products?
I can say from the US FDA side, I mean, that N of 3 golden number often gets accepted, and that also includes the successful batches in sequence, not just the number of batches. But that's, again, a generalization, and depending on your product, the indication you're developing in, there are case-specific differences. And again, that would be part of the negotiation that would occur with the FDA in the appropriate meetings at the appropriate time in development.
Okay, great, thank you. So I think we're exactly at time right now. So I want to thank you again to our speakers, Mike and Emmanuelle, for taking the time out of your busy days to be here. And thank you to the audience as well for joining us and for your questions. So with that, I would like to officially wrap up the ACell webinar series. Thank you again to those who have contributed one way or another to the project, both the document and the webinar series. And thanks to those who have watched and participated in the webinars as well. As a follow-up, we'll be sending a survey, and we appreciate any feedback you may have on the project. And a lot of the recording will be available on the webinar website as well. So thank you again. Have a great rest of your day, everyone.
Thanks, everyone.
Thank you.